with Beckman Coulter, and today the moderator here in this webinar. Well, we're pleased to bring you this session by Dr. Joshua Robinson, who is a senior application scientist at Beckman Coulter. Josh received his Bachelor of Sciences in both mathematics and chemistry from Purdue University. For his PhD, which he did in physical chemistry, he went to Stanford University. Josh's work has focused on developing biocompatible carbon nanotubes, graphene and quantum dots for in vivo tumor imaging and photothermal therapy. Now, before Josh starts, some notes and a few housekeeping items. The presentation will run for about half an hour. We will then open it up to the audience for a moderated question and answer period. You can submit your questions at any time during the presentation just by clicking the little red Q&A widget in your console. We'll try to answer as many of those questions as we can. During the presentation will be a couple of poll questions that appear on the screen, and we hope you'll participate by clicking your answer choice. We also encourage you to download materials from the green resources widget in the console for more information. So please be free to share information about and also this event with your friends and peers on Facebook and or LinkedIn. For your information is that a recorded session of this webinar will be available on demand following the event. One final item before we start. So please, we ask you to do that, really do that. Fill out the 30-second survey at the end of the presentation. This will help us to bring you more webinars which are really relevant for your research, so would help us to finding the right topics and ways to do it. So I will now hand over to our speaker, Dr. Joshua Robinson. Please, Joshua, your turn now.
Thank you very much for that introduction, Dr. Earhart, and thank you all for joining us today for this webinar. We'll be discussing analytical ultracentrifugation and nanoparticle analysis. And for the astute observer, you might notice the small spelling error on the title slide. This is just to keep your attention, as there will also be a few pop quizzes throughout the presentation just to make sure you're awake and with us. A brief outline of the talk. We'll very quickly go over analytical ultracentrifugation theory. This is a very brief look at the theory and doesn't really do it justice, but there are many resources out there, both on the Beckman Coulter website and at the web at large, to be able to learn more. Then we'll discuss deconstructing nano-constructs and how analytical ultracentrifugation has been used in recent literature to really probe the underlying structure of nano-constructs, nanomaterials made out of several different nano species. We'll continue on with general tips and tricks for nanoparticle sample preparation, as well as our current research at Beckman Coulter, and then just a few future perspectives. So for our first poll question, we'll give you about 30 seconds to answer. Just want to kind of know what your preferred technique for nanoparticle sizing is. Just kind of imagine the Jeopardy music playing in the background here. All right, well, that was enough of a break for me to grab a quick drink of coffee. So now let's see what the results are. And by far and away, dynamic light scattering is the preferred method. Now, let's see where analytical ultracentrifuge fits in with these other methods. Electron microscopy, dynamic light scattering, static light scattering, and analytical ultracentrifuge all have a 1 nanometer lower limit, so they're able to probe down to the smallest possible nanoparticle species. One advantage that light scattering techniques and analytical ultracentrifuge have over electron microscopy is population statistics. You're not just looking at a few thousands of particles to relate the entire sample, which may contain upwards of 10 to the 12th or 10 to the 15th particles. But the one advantage that electron microscopy has over light scattering techniques is it has excellent multimodal resolution. This is also something that analytical ultracentrifuge has as well. It has sub-nanometer resolution. So if you have a species that's, say, 5 nanometers and a species that's 6 nanometers, you are able to easily differentiate them with analytical ultracentrifuge, or AUC. And finally, you can also derive the shape factor from AUC, which is just kind of an added benefit. So in basic centrifugation theory, when you have a particle undergoing a centrifugal force, there's really 3 forces in balance. There's the centrifugal or gravitational force that results from the rotation of the centrifuge. This is related to the particle mass and the rotational acceleration of the centrifuge. Larger particles will sediment more quickly. There's also a buoyancy force that's an opposing force to the centrifugal force. This is also known as the Archimedes force. It can be thought of as the solvent molecules are displaced by the sedimenting particle. They have an opposing force. In regular day-to-day life, you can imagine it as if you push a block of wood down into a bathtub, there'll be a strong opposing force from the more dense water molecules pushing back against the less dense wood. And finally, there's a frictional force or frictional drag that occurs as the particle sediments at a given velocity. It'll experience some drag, and this is related directly to the particle's shape. The more aspherical a particle is, the more frictional drag it will incur. Now, in analytical ultracentrifuge, there's actually optical measurements of the particles as they sediment in the centrifuge cell. This is done with either absorption optics or interference optics, but really the output for both is the concentration of the particles at a given radius in the cell. By determining this concentration versus radius and plotting it over time, you can derive the sedimentation rate, the diffusional coefficient, and the frictional ratio, which is related to the shape. Now here's a visual overview of what the absorption optics look like. On the left is a schematic where you see a xenon lamp light source, comes up, hits a diffractional grating, which chooses a wavelength somewhere between 190 nanometers up to 800 nanometers. Now this light is then diffracted down, passes through the cell, but first goes off a reflector, which reflects about 1% of the incident light. And this is to calibrate the amount of light that's going through the cell at any given time, because it's very important for absorption optics to know the exact amount of light that's going through the cell. Then this light passes through the cell, and you see on the lower right-hand side we have a schematic of what the AUC sample cell looks like. There's a sample container, and then there's a reference container. Both contain roughly 400 microliters of sample with a path length of 12 millimeters. There's also a 3 millimeter path length cell available. And the reference acts as basically a background to the UV vis. So when you're taking your wavelength measurement, the reference is subtracted from the sample absorption at each given radius, so you're only looking at the absorption due to your particles. The reference is typically the buffer that the particles were prepared in. The data output looks similar to what you see on the top right. There's an absorption versus radius graph. You see 2 meniscus due to both the reference and sample meniscus. You'll see a very flat region, which is indicative of all the particles having sedimented past this region. And then there's the sharply defined boundary region, which leads up into a plateau, which indicates at which point the particles are still remaining in the cell. On a macroscopic level, you can think about it as putting sand in a glass of water, shaking it up, and watching the sand particles slowly sediment down. The sand particles that started at the top of the glass, I should say, are the ones that you're observing at the top throughout. So the boundary region really gives an indication of how quickly the particles at the top of the cell have now sedimented to this location. And this is how you derive the sedimentation rate. Now this is an ideal case in real life data. You see an output that looks somewhat like this. So this is from real data we collected here using nanoparticles. This was fitted with SedFit. It's a free third-party software by Peter Shook that can analyze the AUC outputs, and there's several other third-party softwares available to analyze the AUC outputs. They're able to be run on your PC or laptop. You don't need a supercomputer. And essentially what they do is they fit this Lamb equation, which is a kind of complicated equation which you wouldn't want to solve by hand, to these curves, and it outputs the sedimentation, which is the rate of movement of the boundary, and also outputs the diffusional coefficient. And that can be seen in the spread of the boundary. So as the boundary spreads over time, there's diffusional force from just random Brownian motion, that there's back diffusion of the particles causing this boundary spread. So you're able to derive both terms from fitting these curves. It just takes a minute or two to fit a curve and get out these outputs. So knowing the diffusional coefficient and knowing the sedimentation coefficient, you're able to pull out quite a bit of data about your particles or proteins. If you see here in equation 2, diffusional coefficients inversely related to F. Well, in this case, F is directly related to the particle radius. So when you pull out the diffusional coefficient, you know the hydrodynamic radius of your particle. In equation 3 here, this is the key equation. You're deriving S and D from the measurement. R is constant. T is the temperature, which is very finely controlled in an AUC run. And then if you know 1 minus V bar rho, so rho is the sample, or the solvent density, I should say, and V bar is actually the inverse of the density of the particles. If you're able to derive those, if you know those two factors, you're able to derive the molecular weight of your particle or molecule that you're looking at. So when you're working with AUC, what can you measure? As we mentioned, hydrodynamic radius comes out from the diffusional coefficient. The shape factor comes from fitting these lines. You pull out the frictional ratio. I should mention that all three forces, the buoyancy force, the centrifugal force, and the frictional ratio, in a centrifuge come into equilibrium within a microsecond. So there's no time that they're not in equilibrium during the measurement. You can quickly assess sample purity or heterogeneity. A popular method to do this is by either looking at just the sedimentation coefficients, which vary rapidly with changing hydrodynamic radius, or by plotting sedimentation coefficient versus diffusional coefficient. This is even better and has nearly angstrom resolution at different species in solution. As you saw, we can do molecular weight determination. We do analysis of associating systems. This is more related to protein work, where you're looking at protein-protein interactions, protein binding. And then ligand binding or surfactant interaction, which comes into play heavily in nanoparticles. When you're developing nanoparticles, typically you have a core nanoparticle with an optical or electronic property that you want to exploit. But typically it's not soluble in a solution that you need to be soluble in, or you need to have some sort of targeting ligand in order to direct it to the place you need it to go. So when you put together these nanoparticle systems, they're really nano-constructs made out of a core and a shell, or a core and a ligand and some sort of surfactant. And general measurement techniques don't do a good job of really understanding the relationship. But AUC, as we'll see, can definitively define what the ratio of surfactant to particle is and how they interact. And these are all direct measurements. There's no calibration needed. You don't need to run size standards and compare your data to the size standards. These all come directly. So quick method comparison. There's a few methods that exist to understand the structural analysis of proteins and nanoparticles. Mass spec, NMR, and X-ray crystallography are very popular, but analytical ultracentrifuge is also popular as well, and it's the only one of these techniques that can also look at the interactions between nanoparticles in solution. So the value proposition with AUC, a technique that's actually been around for eight decades at this point, is that it's highly accurate, has very high resolution when resolving multimodal peaks. It's statistically significant because it's looking at all the particles in solution. It's not just counting a few hundred or a few thousand and extrapolating out the data. And it does look at the particles in their natural state. If you're working with particles in solution, you want to study them in solution. You want to see how their properties are in solution instead of putting them down on a substrate and analyzing them. Now here's an example from a paper which we'll get into more later, but 2 years ago, excellent communication, Nature Communication came out where it looked at gold nanoparticles, which was actually the original system studied by Theodor Svedberg, who invented the AUC in the 1930s. They had 7 different gold nanoparticle species ranging in core diameter from 4.5 to 8 nanometers. When examining by TEM, which is the red dotted line, you see that one broad species was detected. But when run through AUC and plotting sedimentation coefficient versus diffusional coefficient, they're able to pick out all 7 distinct species. And it's visually apparent just by looking at the graph. So, quick quiz here, just making sure you're paying attention. Which one of the forces listed here is not the one that particles feel during centrifugation? If you've been even semi-awake, this one should be pretty easy. So we'll give you about 5 or 10 seconds for this question. All right, let's see what the results are. Hey, everybody knows the right answer. Nice job, guys. Yes, the Jedi force is not a force that particles experience, as far as I know. So when you're running AUC, there's 2 different run types you can actually perform. The first is velocity sedimentation. You place your samples in the cell, and you just watch the speed that they sediment over time. These runs are typically shorter, a few hours, up to a day. They're able to pull out almost all the information needed to study nanoparticles, and they're the more popular run method for studying nanoparticles. There's also equilibrium sedimentation, and this is used occasionally for protein work. This is more for looking at the association states of protein. Essentially what you're doing is allowing the centrifuge to run until all the particles are in equilibrium, so the buoyancy force, the centrifugal force, and the frictional force have all balanced out. These runs can take up to a week, are very long and not particularly useful for nanoparticle studies. Now, first type of AUC is the Proteome Lab XL-I, and this is the one with the absorption optics. The wavelength can be tuned from 190 nanometers to 800 nanometers. So if you have a nanoparticle sample that absorbs at a given wavelength, you can tune the XLA to that wavelength. It has sensitivity down to concentrations as low as 5 micrograms per mil. And it can monitor up to 3 wavelengths per run. So if you have multiple species with different sizes that have different optical properties, you can actually look at 3 different wavelengths, tune it to their peak of absorption for different nanoparticles throughout. Now the other optical method is the XL-I, and this uses interference optics to study the concentration of the particles. Essentially, a light source passes through the cell. You see the schematic on the right here. And the reference sample and the particle sample have different refractive indexes due to the presence of the particles. It's somewhat like Young's double-slit experiment where the difference in refractive index causes an interference pattern that shifts as the refractive index shifts with particle concentration. This interference pattern is seen on the detector, and by counting the shift in fringes per radius, you can directly calculate the concentration of the particles in solution. This method works well for nanoparticles that don't absorb light in the visible region, some liposomes perhaps, as well as if you have your nanoparticles suspended in a detergent or surfactant solution and you want to just analyze the detergent or surfactant solution alone without running the nanoparticles in there to get a baseline understanding, XL-I would be able to run that. The XL-I has both absorption and interference optics on the platform, so it still has the same wavelength range for absorption optics. It's able to determine molecular weights down to 100 Dalton. The concentration range for interference is slightly higher than that for absorbance, so it cannot go down to 5 micrograms per mil, but it does have a higher upper limit of concentration of 5 milligrams per mil. with the absorption, you really want to have an optical density for your samples, a maximum of around 1.5. So if you were using a 12 millimeter path length cell, you're only able to go up so high in concentration. So final pop quiz, this one's slightly tougher. What's the most popular run method for analyzing nanoparticles? I promise no one's getting graded on this, so if you happen to get it wrong, no worries whatsoever. No one will give you a C- or anything. All right, let's see what the poll results are. And the majority of people did get the right answer. Sedimentation velocity is the most popular for analyzing nanoparticles, and some people are having fun with me and selecting run DMC. So I'd like to think that you're just pulling a joke there. So now we've gone over the theory very briefly. Let's get into some recent literature examples of analyzing nanoparticles with AUC. This has become increasingly popular over time, especially with gold nanoparticles in single-wall carbon nanotubes. Both of these particles are difficult to analyze because they almost always need some sort of surfactant or ligand on the surface in addition to the core gold or core carbon nanotube particle. And both are becoming increasingly closer to reaching the clinical trial stage. I believe gold nanoparticles are actually in the clinical trial stage for biological purposes, for biomedical purposes. And as they reach this stage, the characterization of these nanoparticles becomes increasingly important. Currently, you can string together a few analytical techniques and get a rough idea of the overall structure of the particle and its surfactant and its aggregation behavior. But AUC is able to pull together all those measurements into one. So when a regulation agency, say the FDA, wants you to fully characterize your nanoparticle before you put it in a human being and you understand the aggregation effects and the conformational effects of your surfactant at various concentrations, AUC is able to provide that information. With the core shell gold nanoparticle study, this is the study that was referenced earlier in the presentation from 2011. AUC was used to analyze the surfactant coverage on gold nanoparticles as well as derive the average molecular weight of the gold nanoparticles and confirm it with mass spec. This is a fairly busy slide, but this is all the really cool information that they are able to pull out just from running AUC with these 3 separate gold nanoparticle species that had varying numbers of gold atoms in their core cluster. First thing you can note is that they're able to derive the hydrodynamic diameter of the nanoparticles, and you see the accuracy here was sub 1/10 of a nanometer, sub-angstrom resolution on the diameter of these nanoparticles. And then they're able to estimate the ligand shell density. So how many ligands, they're using thiol linkage to some carbon chain, how many thiol linkages, how many carbon chains are there per gram of these gold nanoparticles? This is able to be determined with AUC, and this is very difficult to do with any other technique. And finally, by deriving the diameter, they are actually able to derive the density of the gold nanoparticles, and from there calculate the molecular weight. This was then confirmed by mass spec, and in all 3 cases for all 3 particles, there was less than a 4% error, and the deviation in error was due to slight asphericity of the particles, which was determined by previous literature and the frictional ratio. Now further in this study, they looked at 7 different gold nanoparticle species in solution. As you see on the left, this is the data we've already mentioned where all 7 species were differentiated by AUC, whereas TEM was unable to distinguish even 2 species. On the right, I thought this was a very interesting study. This was slightly larger gold nanoparticles that had a higher sedimentation coefficient, and they were presumed to be 1 species. When analyzed by sedimentation coefficient and diffusion coefficient, the plot indicated that there were actually 2 species present that differed by less than 0.14 nanometers in size. So 2 species apart by less than 0.2 of a nanometer, just basically 2 atom bond lengths apart, were able to be differentiated, and once again, TEM failed. Now, this is not to say that AUC should replace electron microscopy as a technique, but it definitely could be used as a complementary technique or a confirmation of the results seen from TEM if there is some worry about slight changes in, say, optical effects or in vivo behavior or electronic effects from nanoparticles by slight differences in their size. It's best not to solely rely on TEM for analysis, but an AUC run can give more detailed data. Now that project focused on the core of the nanoparticles. There's also a recent paper that came out in 2010 that looked at gold nanoparticles functionalized with DNA that had various number of DNA bases in the ligand. So the DNA provided solubility to the gold nanoparticles, and it was either 5 bases in length up to 30 bases in length connected to a thiol linker. And they saw a decrease in sedimentation coefficient as the number of bases increased. This makes some intuitive sense. They were also able to use this data to confirm theoretical models that they had of the conformation of the DNA bases in solution. So this was an example of using AUC to look at the conformational effects of ligands in solution, which is very important in understanding when you're working with nanoparticles, the access to active sites. A shift in conformation can shift the number of active sites if you're working with some sort of delivery agent. And AUC is able to at least confirm theoretical modeling. Now, a very good case study for using AUC with nanoparticles is carbon nanotubes. There's a groundbreaking work by Arnold et al. In ACS Nano in 2008 where they studied the effect of carbon nanotubes coated with sodium cholate with AUC. Now, brief background of carbon nanotubes. They are essentially a flat sheet of paper rolled into a cylinder, a flat sheet of graphene rolled into a cylinder, excuse me. The cylinder has a diameter of roughly 1 nanometer and length up to a micron or beyond. They're entirely sp2 carbon, so they're highly hydrophobic, and they will not dissolve in really any solvent. So a surfactant is needed to solubilize them, an amphiphilic surfactant if you're working in an aqueous system, which has a hydrophobic portion which will wrap around the carbon nanotubes and a hydrophilic portion to provide water solubility. The carbon nanotubes, as you can imagine, a piece of paper can be wrapped in several different schemes. Carbon nanotubes can be wrapped up in several different ways, and this greatly affects their optical and electronic properties. So by any method of synthesis, they always have a heterogeneous mixture of 10, 20, 30 different wrapping schemes. 1/3 of the wrapping schemes result in metallic carbon nanotubes that have no fluorescence. 2/3 result in semiconducting carbon nanotubes that have intrinsic near-infrared fluorescence, and each carbon nanotube has a different optical absorption peak. So when synthesized, they look like a black solution, but each individual chirality or wrapping scheme has its own unique color. So you see here on the bottom left is a plot of the absorption spectrum of semiconducting carbon nanotubes, and each spot corresponds to a different wrapping scheme with different excitation and emission wavelengths in the near-infrared. Now, the near-infrared is a very fortuitous region to have fluorescence, excitation, and emission for biological imaging because there's a lack of light absorption and scattering by blood, skin, and fat. So you're able to achieve better depth penetration and higher sensitivity when imaging in this region. It's key, though, to have individually solubilized carbon nanotubes, to have a surfactant that breaks them apart into their individual carbon nanotubes instead of bundling all together, because any metallic carbon nanotube bundled to a semiconducting will actually quench the fluorescence. Now, this fluorescence has been used for in vivo imaging of mice. Applied with principal component analysis of video rate imaging, organ analysis and identification was able to be performed. Also, using a surfactant linker, carbon nanotubes were linked to copper-64 radioactive isotope and used for PET imaging of tumors. Now, the surfactant on the carbon nanotubes is very, very important to understand because it greatly affects the pharmacokinetics. You see here on the right, we have a commercially available surfactant known as DisB-MPEG. It has two C18 chains that are hydrophobic, which will wrap around the carbon nanotube, and one hydrophilic chain, which is a PEG chain CCO repeating unit. This imparts water solubility. When coated with the surfactant, carbon nanotubes have a half-life of circulation in mice of 3 hours and a tumor uptake of roughly 4% injected dose of nanotubes per gram tumor. The surfactant on the left is very structurally similar in that it has, each monomer has a C18 chain and a PEG chain to wrap around the carbon nanotubes, but there's several of these monomers stringed together, 90 to 150. And this surfactant, when coating carbon nanotubes, gives a half-life of circulation of 30 hours, so an order of magnitude increase, and also the tumor uptake can be upwards of 20 to 30% injected dose of nanotubes per gram tumor. So the tumor uptake is greatly increased. Part of the reason this could be because of increased surfactant coverage. Part of the reason could be a shift in hydrated diameter of the nanoparticles or nanotubes in vivo because of the very precise size region that is needed to be able to essentially sneak nanotubes into tumors. It's known as the enhanced permeability and retention effect. But you can see that the surfactant and nanoparticle interaction is very important for understanding its pharmacokinetic behavior. What's even more interesting is that when you look at these two surfactants, coat them with carbon nanotubes and compare them with carbon nanotube coated with a protein, human serum albumin, all three activated different arms of the complement pathway in the immune system. So the body's immune response is also tailored to the surfactant choice. Traditional techniques, traditional analytical techniques cannot analyze this, but AUC came to the rescue, so to speak, to analyze the carbon nanotube and surfactant interaction, first shown in this paper by Arnold et al. In ACS Nano. The first step, though, was to do some preparative fractionation of the carbon nanotubes. And this is a key with working with AUC, as we'll get into. You don't want to overtax it, so to speak. When there's 20 or 30 heterogeneous carbon nanotube species based on their wrapping scheme that all have different lengths and possibly different surfactant coverage, that's just too many variables to isolate in one run. So what works best is to actually pull out one wrapping scheme of carbon nanotubes, which can be done with density gradient ultracentrifugation, among other methods. In this way, you see from the fluorescence plot in the top right or the absorption plot in the bottom left, you see a single wrapping species, a single chirality of carbon nanotubes is isolated. The gradient's not particularly fun to run. The gradient steps differ by less than 1% in density, and the run takes quite a bit of time, and it's very painstaking to prepare these tubes. It kind of killed my nerves when I was a grad student, but you are able to pull out a single chirality of carbon nanotubes, and when you run that through the AUC, you're able to get much higher quality data. So this is what Arnold et al. Did. They pulled out a single chirality, the 6,5 chirality of carbon nanotubes, coated it in sodium cholate. On the bottom left, you see the sedimentation coefficients were fit using several different varieties of the Lamb equation. The best fit was the green dotted line. It gave a close fit around 10 to 11 Svedbergs, named after the inventor of the AUC. It's a unit of seconds to the 10 to the minus 13th. And from here, just understanding the sedimentation of the carbon nanotubes and pulling out the diffusional coefficient, you see quite a bit of information in this table that they're able to derive about the carbon nanotubes. So you see they got a sedimentation coefficient of 10.4 Svedbergs, diffusional coefficient. And from there, these are the most interesting pieces of data in my mind. They're able to pull out the anhydrous diameter. So what's the diameter of the carbon nanotube with the surfactant on it, both anhydrous and fully hydrated? So what's the diameter, true diameter in solution? They're able to derive the length of the carbon nanotube in solution, which is very important in my mind because for years I had to calculate the length of carbon nanotubes by putting them down on an AFM substrate and counting dozens and dozens and dozens of carbon nanotubes, but AUC is able to derive that information in solution without having to rely upon electron microscopy. And finally, they're able to derive the exact number of sodium cholate molecules per length of carbon nanotube. So now you're getting an idea of how many cholate molecules are there per nanometer of carbon nanotube. What's the coverage percentage? How much is bare carbon nanotube? How much is surfactant? And this can lead, I believe, to more precise in vivo studies of carbon nanotubes and generally nanoparticles as a whole. Now, one theme with AUC is that improved sample preparation will lead to improved results. So an excellent result just was published earlier this year in March in ACS Nano again by Jeffrey Fagan out of NIST, and their group did chirality separation, so they separated one wrapping scheme, and then they did a second length separation step. So this two-step process led to much sharper AUC curves seen here in figure A and B, and led to, once again, a sharper sedimentation coefficient with lower standard deviation. From here, they looked at multiple surfactants on carbon nanotubes. So they took sodium deoxycholate as a starting point, which is just slightly different from sodium cholate and just 1 point different from sodium tauryl deoxycholate, and looked at the anhydrous diameter for all three. They also looked at the surfactant coverage for all three on the carbon nanotube. So they saw that there was slight differentiation between these three surfactants, and they were able to determine the coverage percentage. And one technique they used is very useful for nanoparticles because to derive all this information, it's important to know the partial specific volume of your nanoparticle, and this is not readily available for most nanoparticles. But what can be done is by running nanoparticles in various density solvents. A typical approach that protein AUC users will know very well is by using H2O and then heavier D2O, and by mixing various fractions of this, you're able to alter the solution density by plotting the sedimentation and the viscosity versus this solution density. You're able to basically extrapolate the line to zero, which indicates that the sedimentation times viscosity is equal to the solution density. So you now know the density of the nanoparticle, and the partial specific volume, which you need for all the molecular weight calculations and so on, is just the inverse of this number. So that's a useful technique to keep in mind when running nanoparticles in AUC. Now, for preparing nanoparticle samples, as we mentioned, it's very important to try to control the physical variables. If you're working with a nanoparticle that you're planning on centrifuging before using it for its final purpose, you should do the same centrifuge before you run AUC. Just use AUC to analyze your final product. You want to prepare a proper reference buffer. Typically what's done is by dialysis into the buffer of choice. So you want to study gold nanoparticles in a PBS buffer, just dialyze 8 times in PBS, and then pull your wash for the final dialysis and use that as your reference buffer. And finally, to get good data, it's best to limit the number of components. You'll see here on the next slide what we mean by that. On the right here is a graphene sheet coated with 2 different surfactants attached to a fluorophore and a targeting ligand. And this is just too much variables for the AUC. You really want to limit it to 2 or 3 variables to understand their interaction fully. You see left, we have a nice happy face for someone who did a density gradient ultracentrifugation separation of gold nanorods that had some impurity of gold nanospheres before running in AUC. So if you wanted to study the gold nanorods, it's best to first do density gradient ultracentrifugation, preparative centrifuge to isolate the gold nanorods for study instead of having to take into account the gold nanosphere impurities. So at Beckman Coulter, we've been doing some research into using AUC for nanoparticles. And one of the areas we're working on is looking at impurities in single-wall carbon nanotube samples. Carbonaceous impurities are difficult to quantify, and one of the reasons is that some of the impurities happen to be, instead of single-wall, they happen to be double-wall or multi-wall carbon nanotubes, which instead of just one sheet of graphene rolled up, they're multiple sheets rolled up. They have very similar physical properties, same length, slight increase in diameter, but the optical and electronic properties are greatly altered. Now, in typical electron microscopy, they're difficult to distinguish. By light scattering techniques, dynamic light scattering can't tell the difference between double-wall carbon nanotubes and single-wall carbon nanotubes in solution. But you'll see with AUC, we were able to solve this issue. So what we did with preparing the single-wall carbon nanotubes was using a Optima Max tabletop model centrifuge, we were able to prepare water-soluble carbon nanotubes without aggregates in 2 minutes of ultracentrifugation. Typical centrifuge times for carbon nanotubes take an hour to 6 hours at 22,000 G. By running for 2 minutes at 131,000 G, we were able to remove the aggregates. The one thing that just saved my life, and I like to thank fellow researchers, Asmita Patel and Ruth Zhang, for teaching me the techniques for this and preparing this. They used the Biomex 4000 workstation, which is an automated liquid handler, to prepare the density gradients to isolate the single chirality of carbon nanotube species. This was much quicker than I was able to run, spared my nerves, and was able to have much better reproducibility between each centrifuge run. Finally, we ran the density gradient ultracentrifugation on the Optima XBN floor model ultracentrifuge by centrifuging at about 300,000 G, 288,000 G for 32 hours. We're able to isolate the single chirality species. I really need to pick up a new camera. My iPhone does not do this justice, but you do see on the right that the black solution of carbon nanotubes before the centrifuge and density gradient was able to turn into vividly colored individual chiralities of carbon nanotubes. We once again collected only the 6,5 carbon nanotube. So here, using the Biomex 4000, or Biomex, I believe is the plural for that, we were able to create this density gradient for the carbon nanotube separation. And we compared this to what can be done manually just by using food-colored solutions of different density gradients. We put them manually by hand in the tube, compared that with the Biomex 4000. We saw there was no difference. When we ran them on the centrifuge, there was no difference. The only difference is I wasn't struggling for hours at a time to prepare this density gradient. We could just set it up in the machine, about 12 minutes per tube. You walk away, do other work. So I believe this is kind of the next step to the industrialization of nanoparticles, especially ones like carbon nanotubes where density gradient ultracentrifugation is a necessary step. Automatic liquid handlers like the Biomex 4000 make this scale up possible. So we collected our 6,5 carbon nanotubes, confirmed that they had the 2 peaks of absorption that we were expecting. We also prepared a solution of double-wall carbon nanotubes with a length fractionation to kind of limit the heterogeneity of the sample. And then we loaded these into AUC cells. You see the pictures here. We had just pure 6,5 carbon nanotubes in one cell, just pure double-wall carbon nanotubes in another cell, and the third cell there is a 50-50 mixture of the two. Now when we ran on AUC, it was readily apparent that double-wall carbon nanotubes had a much quicker rate of sedimentation compared to single-wall carbon nanotubes. This was a sedimentation velocity run, 27,000 RPM. The AUC will go up to 50 or 60,000 RPM. We chose 570 nanometers as the absorption peak to study because that is the peak of the carbon nanotubes, single-wall carbon nanotubes. Controlled the temperature, and so now we have a baseline for both species. When we ran the 50-50 mixture, it was very visually apparent that there are double-wall and single-wall carbon nanotubes mixed together. And by plotting the sedimentation coefficient, we see an easy differentiation between single-wall carbon nanotubes with sedimentation coefficient around 10 to 11, as the previous publications had shown, and double-wall carbon nanotubes with sedimentation coefficients upwards of 70 to 80. And this indicated there may have actually been some bundling of the double-wall nanotubes at more concentrated species. When we plot the sedimentation coefficient for just single-wall carbon nanotubes, the plot in the top left here, we see average sedimentation coefficient of around 11 Svedbergs. This is not as tight as the recent study of NIST, but we didn't do the length fractionation step, and for future work with more time we will be performing that as well. With just single-wall carbon nanotubes, you see there's really no species that have sedimentation coefficient above 20 Svedbergs. With double-wall carbon nanotubes, the peak is between 75 to 80 Svedbergs, and there's a number of species, so there probably is some aggregation of one double-wall nanotube aggregating with another one due to poor surfactant coverage. And both of these were coated with sodium cholate, 1% sodium cholate solutions. But you see there's really no species with sedimentation coefficient less than 30 Svedbergs. So we chose 30 Svedbergs as the cutoff between single-wall and double-wall. When doing the analysis by sedimentation coefficient, the accuracy was within 1%. So 50-50 by absorption mixture of double-wall and single-wall carbon nanotubes gave a 49.6% by 50.4% mixture according to sedimentation coefficient. Similarly, we ran this at different mixture compositions of double-wall and single-wall carbon nanotubes. At both ratios, we were able to accurately determine within 1% of the predicted mixture what it was based on the sedimentation coefficient using 30 Svedbergs as the cutoff. This was also run at two different concentrations to account for any concentration effects that do occur with analytical ultracentrifugation. So to wrap up the talk, just kind of briefly outline some future perspectives. So as nanoparticles begin to enter the biomedical space and enter into clinical trials, better characterization is going to be needed for FDA and the various regulation agencies throughout the world. Furthermore, as carbon nanotubes, gold nanoparticles, graphene, quantum dots, liposomes become scaled up at the industrialized scale for commercial use, as they're being prepared on larger scales, you're going to need more rapid, more quantitative analytical techniques. You can't run 8 different analytical techniques at the end of a batch every single minute to be able to make sure that your process is working well. It'd be best to have one single reliable technique, and AUC, in my mind, is really one of the most powerful techniques to fully analyze nanoparticles. And finally, as the nano-bio interface becomes increasingly popular between an inorganic core molecule and some bio-related molecule, a peptide or a DNA or protein fragment, the understanding of how these interact can be studied by AUC, and I believe that will also be a powerful tool. So in conclusion, AUC is a powerful technique for analyzing the composition of nanoconstructs. It really provides unique information that cannot be gathered from any other particle characterization technique. And when you're preparing your samples, just remember GIGO. If you put garbage in, you're going to get garbage out. So it pays to do some preparative work of your nanoparticles before running them through AUC. At this point, I'd like to acknowledge many people from the Beckman team who helped out. Matt Rania, who gave me the chance to work on this project, Esmita Patel and Ruth Tang, who helped quite a bit with the automation, and Esmita helped quite a bit with understanding of the centrifugation. To get up to speed on AUC, it helped to have some very good trainers. So Professor John Bergner, Irby Purdue, Kate Chen, and Bill Hetzel all helped quite a bit with training me on the AUC. And Jennifer and Lindsay helped make sure this webinar ran smoothly, so that's much appreciated. So now we're going to take questions, but very quickly, if you could answer a short 30-second survey, give you about a minute for this, and then we'll get to any questions you may have. Thank you very much. So I've had a chance to run through the survey. At this point, I'll address several of the questions that were asked. First one here is, how can I know the shape of the nanoparticles by AUC? And this comes out actually from the fitting in one of the third-party software programs, SedFit, Ultrascan. They determined the frictional ratio, or that frictional drag that particles experience while sedimenting. And this is directly related to the asphericity of the particles. So a frictional ratio of 1 indicates that you have a completely spherical particle. Many proteins have a frictional ratio of 1.2 to 1.5, whereas a rod-like particle like a carbon nanotube will have a frictional ratio of between 5 to 10, indicating extreme asphericity. Another question is, how do you estimate the ligand shell density by AUC? This is more mathematically involved, so it's not really appropriate to go over all the math right here, but needless to say, when you understand the density of your core particle and you understand a rough density of your ligand particle, you're able to essentially solve for what part of the particle is due to the core and what part of the particle or what percentage of the particle is due to the ligand, and from there you're able to get the ligand shell density. A question here is, could you measure 5 nanometer and 300 nanometer particles in one run? Doesn't make sense to do an experiment on such a broad sample. And when you run particles that are this far apart in density, they're going to have vastly different sedimentation coefficients. So really, the best way to do this is by varying the run speed during the run to start off at a very low RPM. Around 3,000 RPM is the minimum. So this is what I've used for 300 nanometers, is to start at 3,000 RPM, monitor the sedimentation of the 300 nanometer particles first. Then later in the run, ramp up the speed so you can begin to see the sedimentation of the 5 nanometer particles. The question is, what is the source of our single-wall carbon nanotubes? So this is a carbon nanotube person asking this question. Are they HIPCO, COMOCAT, arc discharge? Typically, in the other studies, I believe people used COMOCAT, cobalt, molybdenum, catalyst carbon nanotubes, because they have the highest percentage of 6,5 carbon nanotubes. In our study, we actually used HIPCO carbon nanotubes, essentially because that's the type of carbon nanotube that I have the most familiarity with. Question is, what's the limit of detection of both components and mixture of single-wall and double-wall carbon nanotubes? And that's actually something that we are currently studying. It seems possible to pick out impurities down to 5%, and we're going to be continuing research to see if we can pick out even lower limits. So at this time, I'll answer all the rest of the questions offline, but I'd like to thank everyone for their time, and I hope that you have been somewhat interested by AUC by this presentation, and there's quite a bit more information that you can find on the web. We'll also be having an application note forthcoming that you will receive in your email for attending this webinar in the next few weeks that goes more into depth into our nanoparticle nanotube characterization techniques we're doing at Beckman Coulter. So thank you very much for your time. Have a good day.